evening, everyone. My name is Himanshi. I'm from BSB Department of IIT Kanpur, and I'm a PMRF PhD candidate. So we'll be discussing about week 9 session and the assignments, previous year assignment, obviously, not the current year assignment. So I guess I'll wait for 3 to 4 minutes more so that more people can join, and then we'll continue with our discussions. Okay. I guess we can start, right? So yeah, if you guys have went through this week's lecture, so you know that this week was completely dedicated to uh, optical system, right? Eye and the eye structure, how we are sensing light, how we are seeing things around us. So if you remember our last week's discussion, you remember that we discussed about olfactory system and auditory system, right? So both olfactory and auditory system, during the session, I guess I have mentioned that we are not predominantly dependent upon our auditory or olfactory system, right? On the other hand, certain animals are very, very much dependent upon their olfactory system for making decisions, correct? And why so? Because we are dependent upon our visual system, correct? So we see our surrounding, then certain decisions are made, and that is how we proceed with our work, correct? So we are completely dependent upon our visual system. But that is not the case always. There are certain conditions in which our visual system doesn't work properly. For example, a person can be blind since birth, or they, during their lifetime, they met an unfortunate accident due to which now they cannot see, or say due to age, as we'll see in today's discussion and certain questions, which actually focuses on age-related detrimental effects on eyes, right? So in those cases, what happens? So if you are blind since birth, right, which means your visual cortex is not at all developed because you haven't sensed a single light since your birth. So in that case, sometimes what happens is our other senses become more stronger. As you know, during evolution, what we use stays with us, and what we don't use, it eventually gets deleted from us, right? Or from the evolution. So if your visual system is not uh, there since the birth, indicating that you unknowingly you will be using other senses to the enhanced level, correct? It can be either a auditory system or a olfactory system or anything for that matter of fact, correct? But for those who are blessed with sight, like who can sense their surrounding via visual stimuli or light stimuli at the end of the day, so there's a complex mechanism associated with it. So if you have gone through this week's lecture, you know what I'm talking about. But if you haven't gone through this week's lecture, don't worry, we're going to discuss not in detail, but somewhat to some extent, that you get an idea about what exactly our visual system does. So the stimuli, that is the light, it is sensed by our which organ? Our eyes, obviously. So what we see is this, just like only the white part and the black part, correct? But if we talk about the detailed structure of an eye, you can see in this diagram that it is made up of so many parts. And this is all things which we have discovered till now, which we have analyzed till now, right? We can see and we can do research and tell about it. So there are majorly 3 to 4 parts, which are further subdivided into more detailed things. So before understanding the structure of an eye, I have kept here as a representative, because obviously I cannot show you the real structure of an eye and keep labeling everything. So before understanding this detailed structure of an eye, I would like you guys to see this very detailed video of the structure of an eye, its parts, not necessarily the functions, because in the coming slides we're going to discuss, eventually with the help of questions, we will discuss most of the functions which are associated with the parts, which you can see here. Okay, so let me go to the video so that we can understand in a deeper sense what eye comprise of, what are its functions. Give me 2 minutes. Okay, here it is. Yeah.
Hi, this is Peter from Anatomy Zone, and in this tutorial we're going to go through the anatomy of the eyeball. Structures responsible for the eyeball is a bilateral and spherical organ which houses the structures responsible for vision. It lies in a bony cavity within the facial skeleton, known as the bony orbit. Take a look at my previous tutorial to learn about the anatomical features of the bony orbit. Anatomically, the eyeball can be divided into 3 parts: the fibrous, vascular, and inner layers. These layers have different structures and functions. Let's start with the outermost layer, the fibrous layer. I've switched over now to a view of the isolated eyeball, with some layers dissected away. The fibrous layer of the eye is the outermost layer. It consists of the sclera and the cornea, which are directly continuous with each other. You can see the shape and continuity of the cornea with the sclera from this lateral view. Their main functions are to provide shape to the eye and support the deeper structures. The sclera comprises the majority of the fibrous layer, approximately 85%, and provides attachment to the extraocular muscles.
Now, I know what medical students say about anatomy or molecular.
The extraocular muscles are responsible for the movement of the eye. Check out my previous tutorial for more information on these muscles. In addition, several vessels and nerves penetrate through the sclera, including the optic nerve, which you can see here posteriorly. The sclera is visible as the white part of the eye. The cornea is continuous anteriorly with the sclera and is transparent and positioned centrally at the front of the eye. Light entering the eye is refracted by the cornea. The vascular layer of the eye lies underneath the fibrous layer. It consists of 3 continuous parts: the choroid, the ciliary body, and the iris, The choroid is a layer of connective tissue and blood vessels. It provides nourishment to the outer layers of the retina. I've switched over to an anterior view showing the whole eyeball to show you the ciliary body, which I've highlighted in green. You can see how it encircles the eyeball. The ciliary body itself is comprised of two parts, the ciliary muscle and ciliary processes. I've sliced the eyeball in half, and we've rotated around to look at the eyeball in section. Let's just review the structures that we have already discussed so that you are oriented to this view. You can see the outer fibrous sclera in continuity with the cornea anteriorly. You can also see the inner vascular choroid and the ciliary body here in green. The ciliary muscle consists of a collection of smooth muscle fibers arranged in three orientations: longitudinal, circular, and radial. When these muscles contract, the diameter of the circular ciliary body reduces in size. Projecting from the surface of the ciliary body are the ciliary processes. These attach the lens to the ciliary body via these fibers, which you can see here, known as zonular fibers. Collectively, the zonular fibers form the suspensory ligament of the lens. The ciliary body therefore controls the shape of the lens. As I mentioned before, when the ciliary muscle contracts, the circular ciliary body reduces in size. When this happens, the zonular fibers therefore slacken, reducing the tension applied to the outside of the lens, allowing the lens to return to a more rounded shape. This process is known as accommodation and adjusts the lens for near vision. Conversely, when the ciliary body is relaxed, the diameter of the ciliary body is greater, which means the zonular fibers are held tight, pulling the lens flat. This optimizes the lens for long distance vision. In addition to adjusting the lens shape, the ciliary body also contributes to the formation of aqueous humor. The final structure of the vascular layer is the iris. This is a circular structure with an aperture in the center called the pupil. The iris is the component of the eye which gives you your eye color. It has two arrangements of fibers which control the size of the pupil: circular fibers and radial fibers. The circular fibers make up the sphincter pupillae muscle, which is innervated by the parasympathetic nervous system. Activation of this system causes constriction of the pupil, which thereby limits the amount of light that can enter the eye. The radial fibers, on the other hand, make up the dilator pupillae muscle, which are innervated by the sympathetic nervous system and cause dilatation of the pupil, thereby increasing the amount of light that can enter the eye. I've highlighted the iris in green and sliced the eyeball in half. Let's rotate around to view the eyeball in section and view the relationship of the iris to the other structures we have covered so far. The inner layer of the eye consists of the retina, the light-detecting part of the eye. The retina itself is comprised of two cellular layers, the neural layer and the pigmented layer. The neural layer consists of photoreceptors, the light-detecting cells of the retina. It is located posteriorly and laterally in the eye. The pigmented layer lies underneath the neural layer and is attached to the choroid layer. It acts to support the neural layer and continues around the whole inner surface of the eye. Anteriorly, the pigmented layer continues, but the neural layer does not. This part is known as the non-visual retina. Posteriorly and laterally, both layers of the retina are present. This is the optic part of the retina. The optic part of the retina can be viewed during ophthalmoscopy or fundoscopy. The center of the retina is marked by an area known as the macula lutea. It is yellowish in color and highly pigmented. The macula contains a depression called the fovea centralis, which has a high concentration of cones, which are the light-sensitive receptor cells which function best in brightly lit conditions and are responsible for high acuity color vision. The area that the optic nerve enters the retina is known as the optic disc. It contains no light detecting cells and is therefore referred to as the blind spot of the retina. The central retinal artery enters at this point, and its branches are visible at this point when viewed during fundoscopy. There are a few remaining structures to discuss to cover the basic gross anatomy of the eyeball. There are two fluid-filled areas in the eye, known as the anterior and posterior chambers. The anterior chamber is located between the cornea and the iris. The posterior chamber is a small chamber located behind the iris and anterior to the lens and suspensory ligaments. The two chambers are continuous with one another via the pupillary opening. The chambers are filled with aqueous humor, which is a clear plasma-like fluid that nourishes and protects the eye. The aqueous humor is secreted first into the posterior chamber and flows into the anterior chamber via the pupil, and is then absorbed into the canal of Schlemm, also known as the scleral venous sinus. This channel encircles the eye, lying at the point between the cornea and the iris. You can see this highlighted in blue on this model. If the drainage of aqueous humor is obstructed, the intraocular pressure will rise, a condition known as glaucoma. The lens separates these two chambers from the vitreous chamber, which fills the eyeball with a transparent, gel-like substance known as vitreous humor or vitreous body, extending from behind the lens to the retina posteriorly. So that's an overview of the gross anatomy of the eyeball. In the
I know it was too much to take uh, into uh, from a single video, but don't worry about the detailed things which were there. You don't need to focus on that much. But I preferred that video because it gave you a holistic overview of the anatomy of an eye. In the lectures as well as in figure, we see things in a segmented way, correct? So we need to see everything as a whole. For example, if I ask you about the type of ciliary muscles, correct? So obviously, with your information, you will directly say that these are smooth muscles. And from the previous sessions, we know the characteristics of smooth muscles. We know things which are associated with smooth muscles, right? So everything will be carry forward to whatever we are studying, correct? For example, if you are talking about lateral or posterior sides, so in the first week itself, we have uh, uh, done, we know what are the segments called, like spatial arrangement of our anatomy. We know what is posterior, what is lateral. So all those things were basics for you guys to understand basic anatomy of complex organs. So you don't need to forget whatever we have studied in first week, second week, or third week. You need to carry forward it, correct? So this will give you enhanced understanding of things. For example, now you know what lateral is, correct? Now you know what posterior is. So after viewing this video, which is, I apologize, it was a very long video, but it was necessary so that you get the complete picture of an eye, correct? So if we then hover over this image, now you will understand better and you will have a clear image of what I'm talking about right now. So if we talk about the posterior part of the eye, the posterior cavity or the front here, which is facing, which you guys are facing and everyone faces. So you know what vitreous chamber is. It is filled with transparent gel-like substance, right? Then you know what canal of Schlemm is, what is the requirement of this canal, and if there is blockage in this canal, what is the disease associated with it. That was glaucoma, right? And glaucoma is both genetic as well as age related. Okay, so in glaucoma, what will happen? The channel through which the aqueous humor is circulated and the pressure is maintained, it is blocked. It will lead to the increased pressure in the eye, which will lead to severe pain. Correct? So it's a very painful condition to have. Then we have sensory ligaments, which will control the thickness of the lens, right? Then at the end of, then we have lens through which we are letting the light pass through. We have cornea, which is the non-vascular structure in our body. Then we have iris, pupil. I hope now you have a clear image of what these things are. Now, if we talk about the anterior cavity, it has posterior chamber, anterior chamber. Again, these are very small chambers. You can go back to the video and you can make your own notes and description, and from your understanding what you are able to get from the video as well as from the lectures of Professor Manik Das. He also has described the structure of an eye in a very detailed manner. So I request you guys to go back to the lectures. Then we have suspensory ligaments, then ciliary body. Ciliary body will control the thickness, correct? Then if we talk about the lateral side of the eye, then it will be sclera, choroid, retina, right? We know now what these things are. We know what optic nerve is. We know what blind spot is. What is the reason that we have blind spot? In the coming slides, we will see what exactly a blind spot is, correct? Then we have all the like vasculature and everything else. So you can make this figure more detailed based upon your understanding. So the best way to learn the anatomy of any organ is to first read about it, or if you want to read about it, first read, then go search a very detailed video, because nowadays we have a lot of information available on the internet, right? So first read, understand whatever you can understand from your reading. Go to the videos which are there, which help you understand in a far better way what you have already understood, and then draw. Correct? In biology, you need to draw, especially in the anatomy or physiology, right? So draw from your memory, whatever you have understood and whatever you remember. Then again, go back to your books. Now see what you have missed. Correct? So in the first attempt, you will be able to cover, I guess, 30% of the major structures. You won't be able to get the detailed information of all. For example, if you won't be able to get maybe sclera, choroid, everything, but you will definitely get retina, right? You will definitely get lens, you will definitely get cornea, iris, and pupil. You might not get ciliary body, you might not get aqueous chamber or blind spot or optic nerve, all those things. But 30% you'll be able to cover. Then go back to the book, see where you have missed, and then again back to your first attempt figure, then try to fill in the gaps. This is the best way to learn anatomy or structure of any organ. This will also help you in remembering things which will last for longer, because if you remember in our neural system when we were discussing about memory, right? So how the memory is formed, there are certain things which we remember. For example, there are dates which we are very clear about. We remember those dates because those were very important to us. And why they were important? Because they had some either an emotional component to it, or a strong stimulus was made. Correct? That stimulus can be of anything. So similarly, if you practice and practice, things will get embedded in your memory. From primary memory or temporary memory, they will shift into permanent memory. And you know, primary memory or temporary memory is a part of hippocampus. So now you can see, right? You have studied so much about neural system that you can actually connect the dots. So if you practice, why people say that practice make people perfect? It's because we are strengthening the synapses and the neural connections which we made during the early stages of learning. So if you practice and practice and practice, those connections will get stronger, meaning your memory will last for longer. Correct? So that is why I'm saying first go to the book, read, go to reference videos or online material. Correct? Then to pen and paper. After pen and paper, again go back to the books and then pen and paper. So this will strengthen your memory for that particular task. Okay. Now, as we see diverse colors, and if you have went through the videos lecture, you know that we have only RBG, that is red, blue, green sensors. We will see what these sensors are. I don't want to overburden you with all the vocabulary and everything. So you can see here that this is one of the uh, representation of how we sense our light and the whole spectrum of colors, right? So this, if you can see the wavelength, this is the blue cones. Cones and rods, if you don't know what they are, go back to the lectures, watch the videos. And if you know, good for you. I don't need to explain what cones are. Cones are the photoreceptors which senses the color, right? So a red cone, blue cone, green cone sensors are photoreceptors. So the blue cone senses the light of this wavelength, correct? Ranging from 400 to maybe 540, 530, correct? Then this is the green cone sensing. Then this is the red cone sensing, correct? And then these are the rods, which not necessarily sense any colors, or they don't help us in knowing what color it is, but they actually help us in viewing. We will see what I meant by this. So you can clearly see that there is an overlap between all these three things, right? Red is overlapping with green and red, green is overlapping with red, blue, red is overlapping with blue and green, correct? So this overlap section right here, these are the reasons that we are able to see the spectrum of colors, correct? For example, if these were distinctive peaks, let me get the laser pointer. Imagine if these were just independent peaks without overlap. So what will happen? We will be only able to see particular colors and not the spectrum of colors. You can also see the representative spectrum right here at the bottom of the figure. That due to the stimulation of blue cone, we can see colors of all combination spectrum till here, right? See right here. With the green, we can see the spectrum from right till here, starting from here, correctly? And then for red, we can clearly see from till this to this. So this is the whole spectrum of colors which we can see due to the overlap of the wavelength through which the cones are stimulated by light, correct? So eye is a very, very amazing structure. How it works, how it is formed, and all those things are very intriguing, correct? So after giving this brief intro about eye structure, in the coming questions also, we will see certain things and we will come back again and again on these major parts of eye itself, depending upon what property we want to understand about eye. For example, if we are talking about lens, we can talk about how much light or how the light intensity is controlled by our eye, correct? If we want to talk about what are our blind spots, so we need to understand what are the layers present at the uh, back of our eye and what is the reason blind spot exists, correct? So throughout the questions, we will understand different aspects of eye, and I assume and I hope that you guys will be able to connect all the things which we have read from week 1 till week 8, because at the end of the day, muscles we have studied, neurons we have studied, and for example, sensor mechanism, how we sense our surrounding, how the signal is transmitted, how things are getting cross-talked, how the signal is sent from our control system to our periphery. We have read everything, we have studied everything. So I really hope that you guys will be able to connect those things. Okay. With this basic intro, we'll go to our question. Let me go back to a cursor. Yeah. So our first question for today is, yeah. And you know the deal that I'll give you guys 1 minute for each question. This is not necessarily only for the participants in the live session. It is also for people who wanna view the videos later, so that they don't need to pause every time there is a question. So this is for their convenience also. And yeah, with this, we'll start with the first question for today. So what is our first question? Our first question says, pheromones are sensed by which sense of ours? Is it auditory, olfactory, vision, or haptic? I'll give you guys 1 minute for this question as usual. Let me post in the chat box. Yeah, I'll give you guys 1 minute, and your 1 minute starts now. Kindly drop your answers in the chat box. Last 10 seconds remaining. Yeah. The correct answer is olfactory system. I hope you guys know what pheromones are. And if not, same answer. Go back to the video lectures, you will know. And even if you don't know, I'm here to give you some idea about it. So pheromones are substances which are secreted to the outside by an individual and received by a second individual of the same species. Many examples exist in animal, but their role in humans remain uncertain since adults have no functioning which processes pheromone signaling in animals, right? Vomeronasal, I guess we haven't discussed. That is, don't worry about it if you don't know about this particular organ, because it is not in uh, humans, right? So we do not know how human senses pheromones, though there are certain studies, maybe behavioral studies as well as cognitive studies, that we are actually able to sense pheromones, but how this functioning or how this happens, we are not certain about it. But for sure we can sense pheromones uh, knowingly or unknowingly, but it is predominant in animals. So just to give you a brief about what pheromone sensing is and what pheromone is, we can refer to this.
Today's topic is, what are pheromones? Everything you need to know. To put it simply, a pheromone is a chemical produced by an animal that causes a behavior change in another animal. It is often described as a behavior-altering agent, but most people mistakenly think it only induces sexual responses. On the contrary, pheromones can trigger all kinds of behaviors in people and animals, along with sexual behavior. They are called ectohormones because they trigger behaviors in other people, unlike hormones, which stimulate reactions inside the body. Let's look at pheromones in detail and see why they affect the body. Functions of pheromones. Animals typically secrete pheromones for various purposes, including 1. Signaling a food trail. 2. Alarming other animals. 3. Triggering sexual arousal. 4. Warning other female insects to lay eggs somewhere else. 5. Giving other animals a warning to get back. 6. Delineating a territory. 7. Bonding between a mother and her offspring. The first pheromone was discovered in 1959. Called bombicol, it was secreted by female moths to attract males. Scientists believe that insects sense pheromones easier than humans do. Animals, the vomeronasal organ, VNO, in the nose is believed to be responsible for sensing pheromones. Also known as Jacob's organ, the VNO is just a few pits inside the human nose. It is present and active while we are fetuses, but atrophies as we are born. Although you might have read all about certain pills making you more sexually active because of pheromones, it's not really true. Several studies have concluded that humans are not affected by pheromones at all, with all studies that have claimed to prove the effect of pheromones considered dubious. Types of pheromones. There are 4 types of pheromones. 1. Releaser pheromones, linked to sexual attraction. These pheromones cause an immediate response that is reliable but also rapid. 2. Signaler pheromones, giving out the generic odor print. These pheromones provide information to help a mother recognize her child in the animal kingdom. 3. Primer pheromones, taking longer to elicit a response. These pheromones usually lead to the development of reproductive physiology, including puberty, menstrual cycles, and the success or failure of pregnancy. Primer pheromones can also alter hormone levels. 4. Modular pheromones, responsible for synchronizing or altering bodily functions. Modular pheromones are released through sweat. They are believed to influence a female's monthly cycles, and experiments have deduced that they can make female animals feel less tense.
Right? So there are different types of pheromones even which are associated with non-verbal communication and also non-physical communication, right? So there are certain animals which actually completely talk on the basis of pheromones. They sense the pheromone secreted by one individual and then their behavior gets altered. For example, there was a recent study. I'm not sure whether I'm quoting it correctly or not. I'll definitely go and find the source for this and post it in the comment box or in the slides itself. So the experiments were done on mice, obviously, because doing experiments on human is unethical. So mouse models were considered for this. And when the female mice was pregnant, the certain pheromone stimulation was given, and that those pheromones were not from the male who uh, impregnated the female mice. So under these stressful conditions, the pregnancy was actually terminated, right? Indicating that sensing of pheromones is so credible for changing the behavior as well as the whole system of pregnancy or sexual drive or food or sensing like any terror or certain bad things in your surrounding. So pheromone research is actually getting certain traction in recent days because we do not know much about it. We have the organ, as you have seen in the video, we have the organ, but it's in the vestigial form. It gets atrophied as we grow old. So does this mean that we do not sense pheromones, or the alternate hypothesis can be that its function has been overtaken by some other organ or some other sensory system of ours? Okay, so most of the things are still in the dark, and we researchers are trying to figure it out, especially with respect to the humans. Correct? So it's a very interesting topic to research. If anyone of you is interested, do give it a try. Yeah. So our second question for today is, yeah, which of the following is not a cell type found in human eye? Is it bipolar cells, ganglion cells, endocrine cells, or goblet cells? You guys have 1 minute for this question. 1 minute. Yeah, you guys have 1 minute for this question. Kindly drop your answer in the chat box. And your time starts now. And same suggestion, if you know the correct answer, good, but you should also know why other options are wrong. This will give you more complete picture of how much you remember and where you guys are lagging. Last 10 seconds to go. Yeah, the correct answer is goblet cells. And if you do not remember the types of cell, it's because you guys haven't seen the video lectures. So please, please, please watch them. Um, so why goblet cells are not the type of cells which are found in eye? Because goblet cells are found in intestine. So goblet cells are a type of intestinal mucosal epithelial cells. We haven't learned about it. Don't worry, we will in the coming lectures, don't worry. So the primary function of goblet cell is to synthesize and secrete mucus. These mucins help neutralize the acid produced by the stomach. They also help in lubricating the epithelium for the easier passage of food. So these are not associated with eyes, these are associated with the intestine. This is all you need to know till today. We will cover in the coming slides. But how are bipolar ganglion and endocrine cells part of human eye or part of an eye? We'll see. But before that, even goblet cells are very special type of cells. So this is the representative structure of a goblet cell. You can clearly see that there's a elongated cell with a nucleus and all the organelles associated with it. But a characteristic feature of a goblet cell is these microvilli, right? You don't often see such microvilli in the epithelial cells. So this is a unique feature which is associated with the goblet cells only. Now talking about the bipolar ganglion and endocrine cells, where they are and what are their functions. Functions we're not going to discuss here. For that, you need to go back to the lecture slides. You will get some idea what these cells are, but I'll give you a certain hint and their orientation. So if you see here, when light is coming, right? So you can imagine and put yourself, because your light will come from this side. If you're sitting like this, your light will come from this side. So when the light is coming, you can see the first layer which the light has to encounter is the layer which is formed by the ganglion cells. The ganglion cells are in contact with second layer of cells called bipolar cells. These bipolar cells are further in contact with the photoreceptors, that is rods and cones. So we have studied about rods and cones in detail because they are the one which are helping us in seeing, but they are not directly sensing the light, correct? They are sensing the light, or in this case, the signal which is generated by or stimulated by the light via these layers, correct? Via ganglionic layer, bipolar layer, and there is a amacrine layer also, but here it is not shown. Don't worry about it. You can go back to the lecture slides. There it is clearly mentioned that even there is a very densely packed layer of amacrine cells also, which, and these cells form a very dense layer which are connected with each other, through which the light signal is being processed. Okay. Yeah. So say next question for today is, yeah, uh photoreceptor, a photoreceptor cell rods detect what? Intensity of light, color of light, both A and B, or none of the above. uh, Wait a minute. Apologies for this. Yeah, I'm sorry for the issue. Yeah. So the photoreceptor cells, rods, detect what? Intensity of light, color of light, both A and B, or none of the above. I'll give you guys 1 minute, though I have already shown you the answer, but still, 1 minute for this question. And your time starts now. Kindly drop your answer in the chat box. Last 10 seconds. The correct answer is intensity of light, right? In the introductory video, we have seen that there are rods and cones. Rods detect something, cones detect something. Both of them together are called photoreceptors. Correct. So I have this very interesting, okay, what happened? Good. Yeah, I have this very interesting paper right here. So what it is exactly, let me show you guys. So this is a review article kind of thing titled Photoreceptors at a Glance. It's an old paper. It was published in 2015, but still I found that this paper covered literally every aspect of photoreceptors. So if you're interested, and it's not also a very uh, technical paper, which that so that you might not able to understand. It's a fairly easy and simple paper. You can give it one read. Obviously, you won't be able to get everything in at a one glance, but it's a fairly thorough paper. So I'll recommend you guys to just go through this. But the interesting thing which I found was this infographic which is associated with this paper. This infographic literally summarizes everything which is known, at least in that time till 2015, about photoreceptors. Correct? So if you guys just zoom in particular sections, for example, if we start here, so you will get a very concise view of photo transduction, how we are sensing light, right? Then you will get to know about the rod's association with it. Then you will see what exactly is the visual cycle. And in the coming questions, we'll see what visual cycle is. Then the structure, like I guess you can now identify which cell is which, right? So this is a rod cell, correct? This is a cone cell, correct? So it's a very, very, very concise, and at one place you can get all the information which is related to photoreceptors. So I found this interesting. Hope that you guys also will find this interesting. If you have even a little bit of free time to go through this paper, it will actually enhance your understanding and give you a deeper, meaningful uh, visualization of our whole and complete optic system from the moment we sense light and from the moment we see things, right? So go through this, and with this, we'll come back to our lecture slides, right? So go do read this review article. So these are rods and cones. Rods detect intensity of light, as you have answered correctly. Cones detect colors. In the coming questions also, we'll see how things are happening. Maybe not in much detail, but you will get an overview of how the light stimulus is converted into electrical impulse, and then how we are sensing things and seeing things. Okay. And again, coming back to how they actually look. So if you look here, this is the confocal image of rods. Okay, this is how actually rods look in the Petri plate under the microscope. To give brief about rods, it's a very concise paragraph from the same article which I have mentioned here. So what it says specifically about rods is that rod and cone photoreceptors are specialized neurons that function in the initial step of vision. These light-sensitive cells lie at the back of the retina, adjacent to the retinal pigment epithelium, a cell layer that is vital for the survival of photoreceptors. Correct? And now connect all this information from the first introductory video we saw today. We saw that the retinal layer was at the back of our eye. Then there was another epithelium cell which was there. And in the lectures, if you see, this epithelium layer is actually providing a cushioning effect and the nourishment for these rods and cones. Okay, so go back to the lectures and obviously the videos and the live sessions. Rod cells are highly sensitive to light and operate under dim lightning conditions. Obviously, because rods are not sensing colors, they are sensing light in general. They are sensing whether the light is present or light is absent. Okay. Yeah. Moving forward. What is? Okay, wait a minute. Okay, yeah. Hmm. Apologies for the inconvenience. Now things will go smoothly. So our next question is, the photoreceptor cells, cones, detect. I know you guys don't need 1 minute for this, but it's my duty. So I'll give you guys 1 minute for this question. So the photoreceptor cells, cones detect what? Do they detect intensity of light? Do they detect color of light? Do they detect both intensity and color of light, or none of the above? Your 1 minute starts now. I know you guys don't need 1 minute for this. The correct answer is color of light, right? Rods detect the intensity of light and cones detect the color of light. In future, if you find yourself getting confused that what senses what, so there's a very basic and common mnemonic is available to learn about what rods detect and what cones detect. So cones start with C, right? So cones will detect colors. C, colors, cones. Okay, and the remaining will be sensed by rods. So I hope that it might help you one day. So the same figure, this is the representative figure of how a rod and a cone cell look like and what are their generic structure. This is how cones look like under uh, very high it's a very high resolution image of cone. It's a SEM image. It's your job to go and search what is SEM. SEM means SEM. It's a type of a microscope, obviously, because we are looking at a microscopic structure. So go and do a search about all the microscope and the high resolution microscopic images, which are nowadays in demand because of the development in the technology. We can actually get very, very clear and detailed images of the smallest organelles which are associated in our cells, and even like very tremendous images are available online also for things which you want to see under microscope. So what are cones? So cone cells function under ambient and bright lighting conditions, in contrary to the rods which worked under dim conditions, right? Exhibit rapid responses to variations in light intensity and are responsible for color vision and high visual acuity. The human retina contains 120 million rod cells and 6 million cone cells, with the latter concentrated in the central or macular region of the retina. Okay, I have this one question for you guys. You can answer in the chat, in the maybe comment section of the video, maybe later whenever you want to answer this. Why there's a huge difference between the numbers of rods and the number of cone cells? The answer is pretty common and obvious if you connect it with the senses, like rods, what rods are sensing, what cones are sensing. But I want you guys to think about it, that why there's a huge difference in the number of cells, because rods are literally 120 million and cones are 6 million in number, correct? So just give it a read and let me know why there's a huge difference in the number of rods and cones. For us, the next question for today is, during an eye transplant, what is transplanted or replaced? Is it the whole eye, the retina, the cornea, or the lens? So you guys have 1 minute for this. And your time starts now. Last 10 seconds to go. Yeah. The correct answer is cornea, because it is pretty understood that we cannot transplant the whole eye, right? Though it is called eye transplant, but we are only transplanting a part of an eye, which is the cornea, which is most of the time affected and leads to the eye transplantation. So there's a very basic flow of how the cornea is transplanted. It's a very simple flowchart of it. So you have a damaged cornea, which is removed surgically. Then the donor cornea is implanted and then it is sutured. To understand this in a better way, here's a small video related with the eye transplant. Okay, let me share it with you guys so that you'll understand what exactly happens during eye transplantation. Though it seems as a easy process, but like every surgery, it has its concerns and it has its things to keep check on, because at the end of the day it is surgery. Though it is common surgery, but still we should be avoid to the maximum getting surgeries, and we should try to get things corrected with medicine or behavioral therapy or those things. And at the last resort, a surgery should be considered as an option. If there is no other option available, then we should go to the surgery. Okay, irrespective whether it's a easy surgery or a complex surgery.
The cornea is the clear outer window of the eye and is the only clear part of our body. Its function is to allow light rays into the interior of the eye to focus on the retina. Corneal disease is a serious condition that can cause clouding of the cornea, visual distortions, and eventually blindness. Corneal disease may result from infections, injuries, or hereditary diseases. We routinely check for corneal disease by examining your eyes with powerful microscopes. If the cornea becomes cloudy or distorted as a result of corneal disease, the only way to restore sight may be surgery. Corneal tissue for transplants comes from a donor. With a full-thickness transplant, the diseased cornea is removed, and a full-thickness donor cornea is sutured into place. Return of best vision is gradual and may be recognized in 3 or 4 months, while for some patients it may take up to a year. DSEK is a relatively new surgical technique that can help some patients who previously required a full-thickness corneal transplant. Both surgical procedures require use of a donor cornea, but DSEK replaces only the diseased posterior section of the cornea called the endothelium. DSEK may be recommended when corneal disease is limited to the endothelium, as in Fuchs dystrophy, a hereditary condition. First, the damaged endothelium is removed from the diseased cornea and a thin disc of healthy endothelium is prepared from the donor's cornea. The healthy endothelium is then placed inside the eye, where it is held in place by an air bubble. Because of the smaller incision used with DSEK, many people experience improvement in their vision within weeks. Sutures may or may not be used at the surgeon's preference.
Right? So even a minor surgery has certain risks associated with it. So as a last resort, everyone should consider surgery, and also people should not shy away from surgery if it's absolutely necessary, right? So just a side note. Moving to the next question. Yeah. So next question for today is, cataract leads to blank, and during surgery, it is replaced by blank. So you need to fill in the blanks based upon the options available to you. So cataract leads to blurred cornea, and during surgery, it is replaced by artificial cornea. Or cataract leads to cloudiness of lens, and during surgery, it is replaced by intraocular lens. Or cataract leads to retina damage, and during surgery, it is replaced by intraretinal retina. Or last, cataract leads to pupil dilation, and during surgery, it is replaced by Botox. So you guys have, as usual, 1 minute for this question. Drop your answers in the chat box. And your time starts now. Last 10 seconds to go, guys. Yeah, the correct answer is cloudiness of lens and intraocular lens, meaning cataract leads to cloudiness of lens, and during surgery, it is replaced by intraocular lens. Correct? So what is cataract? By now, you know what we mean by cataract disease. You can clearly see that there's a cloudiness in the there is a cloudiness of the lens, right? And why this cloudiness occur? If you have seen the video lectures, you know why this cloudiness is there. Lens, I'll give you a hint, and if you don't remember and you have gone through the lens, lens is made up of a certain protein, right? And that protein is transparent in nature, due to which we can see, right? That protein is misfolded due to unknown reasons. The research is still on, and we do not know why this happened, and especially why it is related to age. So due to the misfolding of that predominant protein, there is cloudiness, and the protein is no more transparent, leading to the inhibited or limited light passing. Okay. Now, to understand this, there's a small video associated with this so that you guys can understand in a better manner. Let me skip the ads and then I'll share. Yeah. Here it is.
If you have blurred vision or other complications due to a cataract in the lens of your eye, your doctor may recommend surgery to remove the cataract. The lens is a clear disc that receives light through the pupil and focuses it on the retina to help produce a clear image. The lens is mainly composed of water and proteins. As you age, these proteins may begin to clump together and cloud the lens. This clouding is called a cataract. A cataract can block light from entering your eye and cause blurry vision. Before your procedure, you will be given eye drops to dilate your pupil, and the area around your eye will be washed. In most cases, you will be given an injection of local anesthetic around or behind your eye. You may also be offered a sedative to help you relax. Most cataract surgeries last less than an hour. The most common method for removing cataracts is phacoemulsification. In this procedure, your surgeon will look through an operating microscope and make a tiny incision on the side of your cornea. He or she will make an opening through the capsule of the lens and use an ultrasound probe to break the cloudy lens into tiny fragments. Next, your surgeon will vacuum out these fragments through the incision. Then, an artificial intraocular lens will be inserted. Intraocular lenses are made of plastic, silicone, or acrylic compounds and will remain in place for the rest of your life. Because the incision is very small, stitches are generally not necessary. Vision often improves notably immediately after surgery. After your operation, a patch will be placed over your eye, and you will be observed for a short time to be sure there are no problems, such as bleeding. Cataract surgery is performed on an outpatient basis, so you will likely go home the same day. Since exposure to bright light may initially be uncomfortable, make arrangements for someone to drive you home.
Yeah, correct. Though it looked very scary, but it's not. Imagine if the incision is so small that you don't need any uh, that you don't need any suture or stitches, right? So it looked scary in the video because it was for demonstration purposes how things are done. And why I am showing you guys all this, it's because so that you get an idea about how actually things work. And I actually prefer looking at the videos to understand certain concepts. I do not solely depend on them. I always go back to the books and lecture notes, but for initial understanding and making concepts, you can always check for the videos which are made by authentic sources and which are explained in a way that you are able to get the concepts right. Okay. So our next question for today is, at the blind spot, there are dash receptors, photoreceptors. Maximum, no. Half the density of, or horizontally aligned. Meaning, at the blind spot, there are maximum photoreceptors, or at the blind spot, there are no photoreceptors, or at the blind spot, there are half the density of photoreceptors are saturated there, or at the blind spot, there are horizontally aligned photoreceptors. So your 1 minute for this particular question starts now. Kindly drop your answer in the chat box. Last 10 seconds to go. Yeah, the correct answer is no photoreceptor. So obviously this question didn't need 1 minute, but as the name itself suggests, right, it's a blind spot, meaning there will be no sensing of light. And what are the things which sense light in our system? These are the photoreceptors. So at the blind spot, there are no photoreceptor. So in the introductory video also, we had discussed about certain major components of the structure of an eye or the anatomy of an eye. And there it's a predominant structure. And here is a simplified anatomy and the like labeled figure of the eye itself. You can clearly see that there is the blind spot and these are the retina, right? So we don't have any photoreceptor which are associated at the blind spot. To make information and knowledge more concrete in your head, here is another perspective of how we can see the anatomy of our eye and understand certain basic things, right? And to know how we basically see things and sense light. So with that, let me go to this. Yeah, here it is. Yeah.
Your eyes help you see the world, but you can't see this dot. No, seriously, if you close your left eye, stare at this crosshair with your right eye, and slowly move your head towards or away from the screen, the dot will disappear, revealing the
I would like you guys to try this for not 1 minute. Don't strain your eyes, right? Just close your uh, left eye and see this dot, or you can go cross will be better. So close your right eye, with your left eye try to see the dot, move closer to the screen and see there will be a time when you won't be able to see this plus sign, though it is right there in your vision region. Okay, so I'm not gonna give you guys time for this because we are short on time. So do try this at your home and then you will see what exactly is the meaning of blind spot. Okay.
exact location of your blind spot. The cells at the back of your eye transform light into signals that are sent to your brain, but they are missing right here because this is where nerves and blood vessels connect to the eye. So anything at that point in your vision you can't see, aka your blind spot. If we add a line through the image and you try again, you'll notice that your brain fills in information and makes you think the line is continuous instead of there being a hole in your vision. You also can't see all 12 of these black dots at once for a similar reason. Go ahead and try. Chances are you'll only see a fraction of them at once. The exact point your eye looks at is focused, but your peripheral vision isn't great, and so your brain often makes assumptions for what's there. Because of the consistent gray line pattern, your brain assumes the rest of the image is just like that and misses the black dots until you look directly at them. These two rectangles are flashing out of phase with each other, right? How about now? For most, the rectangles will now seem to be flashing at the same time in phase, but they aren't at all. And yet, if we move these shapes beside them just a few pixels, you'll begin to see them out of phase again. The surrounding area has a direct impact on how you see and perceive things, even if the result isn't correct. Not convinced? Look at these flashing squares. Except what you might not have seen is that the middle square isn't flashing at all. If we remove the outer square, we see it for what it is, a solid color. Now try reading the sentence inside it. Did you catch the extra word? Your brain doesn't always notice mistakes like these because it doesn't affect your comprehension of the sentence, and your brain would prefer to act quickly rather than to be perfectly accurate. Try staring at the middle dot in this illusion. When the picture isn't moving, the colors are clearly changing quickly, yet when the image starts rotating, the color change either seems non-existent or much slower. Our eyes and brain have evolved to see, but our vision makes assumptions based on learning, memory, and expectation, and all of these illusions take advantage of this adaptation. It's an advantage to have rapid information processing, so instead of taking in every bit of detail to be 100% accurate, which would cause a brain overload, your brain makes assumptions. This faster perception allows for faster reflexes and faster conscious and unconscious decisions, an integral aspect of human nature. Much like this halo, if you stare at the center dot for long enough, the brain will simply make it disappear. It makes an assumption that the information is unchanging or unimportant, and as a result, you can't see it. Love tricking your brain with illusions? We've put together
How many of you remember there was this let me remember that it was, I guess, yeah. So do you guys remember this, any of you? right? This dress right here, this caused so much of internet sensation, that what is the color of this dress. So that was our mind tricking us, because our visual system will see what is right in front of us, right? So if we change the source of light and where we are sitting and we are seeing and in what light we are visualizing things, things actually change. So we don't know the absolute exactly how in reality things are, because we are viewing things with certain reference and certain memory and expectation, right? That is how our brain are filling things and help us in being more spontaneous, making decisions quickly, making our reflexes strong. Okay, so this is despite being a drawback that we are not able to see things how absolutely they are, but it's an evolutionary advantage at the end of the day, which has made us quick decision makers, quick reflex, like we have good reflex arc because of that, and we can judge our surrounding in a better way. There are certain animals whose eyes function in a far better way in comparison to humans, but we are the apex predator, right? Yeah. So moving to the next question for today, we have dark current is. So what is dark current? Is it depolarizing, hyperpolarizing, either A or B based on the color of light, or either A or B based on the intensity of the light? So as usual, you guys have 1 minute to answer this question in the chat box. And your 1 minute starts now. And if you don't know the answer, don't worry. Try to remember the terminology which are present here, because every question gives you an opportunity to revise your learnings. So if you don't know what dark current is, but you know that we have studied about depolarizing and hyperpolarizing things, right? So you can always start remembering what is a depolarization and action potential, where you have studied what are the things associated with it. Same with the hyperpolarizing. Last 10 seconds to answer. The correct answer is depolarizing. So what is the dark current? So in the dark, CGMP levels are high and keep CGMP gated sodium channels open, allowing a steady inward current called the dark current. Don't worry if you don't know these terminologies. We're gonna see certain references and then you can easily understand what I mean by the things which are written here. The dark current keeps the cell depolarized around minus 30 millivolt, leading to glutamate release. Light leads to reduced CGMP levels and closure of CGMP gated sodium channels. You might not get everything which I have written here, but you know what are gated ion channels. So if it's a sodium channel, it is a gated sodium channel and it is gated by CGMP molecule, right? So you can decode the name of channels and receptors from their name itself, correct? Now to understand what is dark current, we'll see, because depolarizing is what meant by dark current. We know what depolarizing effects are or hyperpolarization is because we have read all those things in the where we have studied these things.
पहली दिवाली है
During the
इसे क्या दे रहे हो
मेरी साली है
action potential generation, right?
वाओ इतने सारे इंटरनेशनल ब्रांड्स दिन ऑफ द ईयर से सीधा जीजू ऑफ द ईयर
Mintra BFF Early Deals on Ice. Shop now.
This, I'm sorry.
Welcome to 2-Minute Neuroscience, where I explain neuroscience topics in 2 minutes or less. In this installment, I will discuss phototransduction. Phototransduction is the process that occurs in the retina where light is converted into electrical signals that can be understood by the nervous system. It primarily takes place in photoreceptor cells, of which there are two main types, rods and cones. I will discuss phototransduction in rods, although the process is similar in cones. In the dark, positively charged sodium ions flow into rod cells through ion channels that are activated by a substance called cyclic guanosine monophosphate, or CGMP. This influx of positively charged ions causes cells to remain in a depolarized state, leading to the continuous release of the neurotransmitter glutamate. Inside the rod cell, there is a substance called rhodopsin, which is made up of a protein called opsin and a molecule called retinal, which is capable of absorbing light. When retinal absorbs light, its configuration changes, an event that prompts opsin to activate a protein called transducin. Transducin activates a type of enzyme known as a phosphodiesterase, which begins breaking down CGMP. As CGMP levels fall, the ion channels that are opened by CGMP begin to close. Thus, less sodium enters the cell, and the cell becomes hyperpolarized due to potassium ions that regularly leave the cell. Consequently, glutamate release decreases. Strangely enough, this decrease in neurotransmitter release acts as a signal that a light stimulus is present. The rod cell returns to its normal state quickly when activated rhodopsin is inactivated, and a protein called arrestin subsequently binds to it. Arrestin blocks the ability of rhodopsin to activate transducin, which makes the cascade unable to continue. A complex process then restores retinal to its original configuration, making it ready to absorb light once again.
Right, as you can see how we are sensing the light. This is how our rods work, and in the similar fashion our cones also work. So we basically sense the light due to the channels which are CGMP gated. These are sodium channels, and due to the metabolization of the CGMP, the channels are closed, leading to no influx of sodium and increased potassium. And if you go back to your action potential lectures, you will know what this condition is called. It is called depolarization, right? So dark currents are depolarizing state or depolarization current of the eye when our eyes are sensing light.
Of the eye, light-sensitive rod cells contain stacks of membranes, called discs, in which are embedded the light-sensing receptor rhodopsin. Within rhodopsin is a molecule of retinal, which exists in its 11-cis form. When 11-cis retinal absorbs a photon of light, it converts its energy to an atomic rotation around the carbon-11 and carbon-12 bond, forming all trans retinal. This minute change in the structure of a pigment initiates the cellular events leading to vision.
Right. So this was a very small video which told you about the biochemistry of how we are sensing light. So in the first video, we saw how we sense light and the rhodopsin is there, and there's a change in the rhodopsin which leads to the decreased CGMP levels, which further leads to the decrement in the glutamate. The same thing is written here. So I hope now that you have a better understanding of what is written here. and decrease in the glutamate level indicates that a light has been sensed. But the complex process of the rhodopsin, which is a light sensing pigment present in the photoreceptors. So there's a way at a molecular level, at a single molecule level, what is happening? Due to the energy which is coming from a photon or light, there is change from cis configuration of the molecule, from cis configuration of a molecule to trans configuration, which leads to the other chemical and biochemical processes, which finally leads to the decreased glutamate level. So on broader perspective, what is happening? Light is coming, rods and cones are sensing. So how rods are sensing? When a light enters in a rod, there is a rhodopsin pigment. Rhodopsin pigment, there is a retinal small molecule. And what are retinal? Those are vitamin A, right? Reason we say that eat vitamin A for good eye health, right? Or for good eyesight. Because the pigment which is changing from cis to trans is the derivative of vitamin A, correct? So you can see how things are closely related. It's a beautiful way how our eyes work. I'm always mesmerized by the peculiarity and the accuracy of how we sense light and our surrounding via our visual system. I hope that this is interesting to you guys also. Yeah, so next question for today is resting potential for photoreceptors is minus 90 millivolt, minus 40 millivolt, minus 75 millivolt, or plus 90 millivolt. I'll give you guys 1 minute for this question. Kindly drop your answer in the chat box. And your time starts now. Last 10 seconds to answer. If you don't know the exact value of resting potential of photoreceptors, how you can solve this question depending upon the information which you have gathered from your previous lectures, which were related to potential or action potential, right? So if there is a high negative resting potential, what it will signify? It will signify that you need a lot of stimulation, correct, to overcome the barrier, right? If it is extremely negative, there, if you remember, there was a threshold of the action potential that you need that threshold level to cross for the action potential. So if there's a lot in negative value, so you need more stimulation to cross that threshold. And we are here talking about photoreceptors, which are highly sensitive, correct? So you can relate this information that photoreceptors, which are highly sensitive, so do they need a lot of stimulation to cross the threshold value, or do they need less threshold value, less stimulation to cross the threshold value, correct? With this information from your previous knowledge and studies, you can try to attempt this question. So if you have gathered this concept, the answer will be pretty obvious that it will be minus 40 millivolt. And if you don't remember this, you can go back to the lecture slides and you can check what was exactly the reason that we have such low resting potential for the photoreceptors. One reason is this, because they are highly sensitive. Because we are continuously sensing our environment by our visual system, right? So if there's a lot of negative value, there will be lag in perceiving the light and then actually seeing the object. Correct? So there's a small thing which is associated with this. Let me go through it. So unlike most sensory receptor cells, photoreceptors actually become hyperpolarized when stimulated, and conversely are depolarized when not stimulated. This means that glutamate is released continuously when the cell is unstimulated, and stimulus causes release to stop. In the dark, cells have a relatively high concentration of cyclic guanosine 3,5 monophosphate, that is the CGMP, which opens CGMP gated ion channels. And now you know what were these ion channels? They were sodium ion channels, right? So these channels are non-specific, allowing movement of both sodium and calcium ions when open. The movement of these positively charged ions into the cell, driven by their respective electrochemical gradient, depolarizes the membrane and leads to the release of the neurotransmitter glutamate. Unstimulated in dark, cyclic nucleotide gated channels in the outer segment are open because GMP or CGMP is bound to them. Hence, positively charged ions, namely sodium ions, enter the photoreceptor, depolarizing it to about minus 40 millivolt. Resting potential in other nerve cells, if you remember, it was minus 65 to minus 70 millivolt. This depolarization current is often known as dark current. Correct? Yeah, moving to the last question for today. So what the question says, what part of the eye helps in changing focal length of the lens? Is it retina, pupil, cornea, or ciliary muscles? I know you guys don't need 1 minute for this, but still I'll give you 1 minute for this particular question. Yeah, kindly type your answer in the chat box. Obviously, the correct answer is ciliary muscles. And what are ciliary muscles? The ciliary muscle is an intrinsic muscle of the eye formed as a ring of smooth muscle in the eye's middle layer, uvea, which is a vascular layer. It controls accommodation for viewing objects at varying distances and regulate the flow of aqueous humor into Schlem's canal. So now you know what these words are, because we have seen previously in the video that what were these. Before coming to the actual things, how they work. Oh, okay, I guess I got the wrong video. Give me 1 minute. Okay, yeah, here it is. We'll see how a very minute change in the structure has allowed us to see things which are at a distance and which are at nearby to us.
Theory of reciprocal zonular action, and formulates a new consolidated theory of accommodation as follows. The engine of accommodation is the contraction of the ciliary body mediated by the distribution of forces via all elements of the zonular apparatus. The resultant changes in refraction are due to the increased thickness of the lens and the increased curvature of the anterior and posterior capsule. The complex anatomy of the vitreous zonule results in traction on the vitreous membrane peripherally and traction on Weigert's ligament connecting the posterior capsule and vitreous membrane. During accommodation, the ciliary body moves forward and centripetally And at the same time, the posterior zonular attachments, interconnected with the vitreous membrane and the posterior lens capsule, are also stretched, contributing to the shape changes in the lens. During disaccommodation, the lens returns to its original non-accommodative state as a result of traction of the anterior zonules, accompanied by a return of the vitreous compartment and the shape of the vitreous membrane to its non-accommodated state. There is a reciprocal action of the anterior zonules and the posterior zonules. During ciliary body contraction, the anterior zonules lose tension, while the posterior zonules stretch and exert force on the posterior lens capsule, playing a role in shaping the posterior lens thickness and curvature. During ciliary body relaxation, the posterior zonules lose tension as the lens flattens and is pulled back by the increasing tension of the anterior zonules.
So you can see how minute thing, like just a little bit of relaxation and contraction of ciliary muscles leads to the accommodation of our lens for viewing things which are at distance and for viewing things which are near to us. This is a very small representation of when the ciliary muscles are contracted, what happens to the lens, and when the ciliary muscles are relaxed, what happens to the lens. So it is not only the ciliary muscles, though they are the ones which are relaxing and contracting, but the ligaments which are associated with the muscle, they are actually leading to the change in the shape of the lens. It's so marvelous, right? How small change in something enhances and multiplies if things are associated with it, correct? So with this, I would like to thank you all for your patience and congratulations that you are stuck till here, at least like till week 9, and you are continuing learning. With this, I leave you all with the question to ponder upon for this particular week. And the question is, can you think of an easier way how body could have made lens? So if you don't know how we are making our lens, which is the eye lens, obviously, how that process is there, go back to the lectures. It's sir has explained in a very detailed manner how the lens are constructed. So could you think of any other feasible way? Because when you go back to the lecture and see how lenses are made, there's a association of destruction of the membrane, right? So can you think of any other way how we could have made lens which is more energy efficient as well as more optimized? So it's just a thought experiment, give it a try. Okay. So thank you for your time and thank you for your patience.